Hello, my name is Roger Jones, and I'm coming to you from Pensacola, Florida today. It's not steamy here in Pensacola today because it's just a couple days before Christmas in 2018. I have a guest with me today, and I'll introduce him in a minute.
Oh, yeah.
Uh, but what we want to talk about today is something that's been in the news recently and probably needs some clarification. Recently, a scientist in China did something called gene editing on human beings. Uh, and what we want to talk about today is just what is gene editing and what does it mean and how is it related to uh, other kinds of genetic engineering. We have with us today my brother, Alan Jones, who is a professor of biology at the University of North Carolina. And so I'm taking this opportunity to ask him a couple questions to tap into his expertise a little bit uh, about just what are these things that are in the news. So, Alan, let's start with the first absolute basic question. What is gene editing?
Gene editing is a tool we have in a large toolbox that enables us to manipulate the DNA that encodes for all the proteins that do work in our body, in our cells.
Right. So it basically, you mentioned proteins. Can you talk a little bit more about what that means? What is a protein?
Well, proteins are a polymer that fold up in a certain way that can do work. They do they do reactions. They control reactions. For example, the pigment in our eyes or our hair, our our skin. They enable me to move my hands, to have neural transmission for me to think. So proteins are the key things for doing all the work that make up who we are.
So they're the workhorses.
Yeah.
And then how are proteins related to genes then?
Well, these proteins are all very different, and they're made up of units themselves, and it's the order of these units that enable them to have the proper shape to do work. And so these units themselves are encoded in a blueprint, so to speak, which is the DNA that we have. And so and it truly encoded is a great word because there's a series of chemical building blocks. They're called nucleotides, and there are four of them. And the order in which they are in each other encodes that information for a particular protein. So one gene, one protein, for example. We have many genes because we have many proteins.
Oh, okay. So each gene is a stretch of DNA that
Mm-hmm.
encodes a single protein.
Mm-hmm.
Okay. And then,
And it's the order of those bases, those nucleotides.
uh, all right, then what are the subunits of a gene?
Well, so they're nucleotides, and there are four of them. And to keep it simple, we give them letter codes:
Uh-huh.
A, G, T, and C.
Uh-huh.
And so the order of these nucleotides, then, uh, that order in itself, repeating of those four subunits, is decoded into the information it needs to make that protein.
Uh-huh.
Okay?
All right. And then something that's mentioned are base pairs. What's that?
Base pairs, right? So our DNA is a polymer of these nucleotides, but we have two strands of them.
Uh-huh.
Okay, we have a coding strand and a complementary strand. So and they pair together. All right. So as I mentioned, we have A, G, and T and C. Um, they the two strands are pairing together through these bases, and only in G's pair with C's, and A's pair with T's, and so on and so forth, so that you have two strands that are paired the whole length of the way. And so you'll read a lot, and you have in the newspapers is talking about editing the base pairs. Really, what we're talking about is uh, changing not just the nucleotides of one strand, but changing it in both. You're changing the pairing. Does that make sense?
Yes. So let me just say it in my own words.
Good.
You have a gene, and the gene is some stretch of DNA.
Right.
Okay, some piece of DNA, and this gene encodes a single protein. And then you have inside the DNA, you have base pairs, like A and T is a base pair and G and C is a base pair.
You're right.
And these base pairs form the double helix of the DNA molecule.
Right.
And really, what we're talking about here is going into the gene itself and changing the base pairs inside the gene. Is that correct?
That's right. And by changing those base pairs, you're changing the information that encodes for that protein. And so potentially, if you engineer it properly, you can change the protein itself.
Well, all right. So this is fascinating. So how is gene editing related to other similar kinds of things we hear about in the news, like genetically modified organisms and such?
Yeah, that's a really good question. So gene editing, you hear so much of now, is just another tool, as I said, in this toolbox in which we manipulate the genes. And in my opinion, that is on the a whole umbrella of genetic engineering. And we've been doing that, in my opinion, for thousands of years by selecting for traits at the organismal level, by recombining genes from one parent and another, and then selecting amongst the, the progeny, the children, certain traits that we want. And we used the example of dogs, for example. Um, but that's crude. And on the other end of the spectrum would be gene editing, and that is this very surgically precise change of the pairing of those bases, as opposed to just mixing genes together and looking for some change that you might want. In between that is something you've heard about and you've asked here, and that is genetically modified organisms. And that just about, in my opinion, pertains to anything that we modify at the genetic level. But most people think of it as where you put DNA from other sources into. Let's say you wanted to take and take DNA from salmon, the fish, and put it into a tomato. Why you would do that? Well, maybe you're trying to create a tomato that can survive harsh freezing, right? You'll never find that in nature. You'll never find that by taking two different tomato parents and trying to find it. But you could if you knew that the salmon had an antifreeze kind of protein, a protein that provided that tolerance to hard freezing. And if you put it in the tomato, maybe we could make a tomato that can survive, a harsh freezing and we wouldn't lose our crops. Okay. Or maybe you wanted to take genes from an organism that prevent the oxidative or the kind of browning you would have in an apple, and you could put that in. So these are genetically modified organisms, GMO, as you said, in which DNA, foreign DNA, is moved in, or you could take existing DNA and totally remove it. These are not quite as precise as genetic editing, where we started, but it's another tool in the toolbox of genetic engineering. Is that help?
Yeah, let me
Okay.
summarize and
Yeah.
see if I got it right.
Okay.
Um, so we have all these tools for genetic engineering, and the one that's oldest is breeding. Uh, we have farmers breeding various animals to improve their conditions. This is a very coarse kind of genetic engineering because we're dealing with organisms,
Yeah.
and organisms that
And it's not just breeding, right? Because you can get natural changes too.
yeah.
And so you would also be utilizing the natural mutation or the normal background changes in DNA, which occurs from sunlight and exposures to chemicals and things like that. So it's not just taking two parents, it's also using So I believe somewhere maybe in the Old Testament or somewhere 2,000 years ago, they talk about that branch of the olive tree that gave the better olives.
Uh-huh.
And they said, take the olives from that branch, selecting it. And that came by, a mutation that just occurred,
Uh-huh.
right?
Yeah.
Yeah.
Right. So really, at that level, at the breeding, etc. level, we're operating in Darwin's world, where
Yeah.
organisms are evolving.
Yeah.
Then we have a little bit finer tool where we can take genes from one organism that has a particular trait we like and put it in another organism that doesn't have the trait we like, but we would like it to have it.
Yeah. Yeah.
Uh, and so this is the genetic GMO.
So not only is that faster than breeding or
Yeah.
waiting for evolution to create something by natural change, uh, but it is providing us a means to engineer something that may never exist otherwise.
Yes. Okay. And then now we have tools to go into the gene itself. We're not talking about swapping genes from one organism to another. Go into the gene itself and modify the base pairs inside the gene.
Mm-hmm.
And so really what we're doing then is, if I understand this correctly, is we're redesigning a gene.
Yeah, yeah, that's right. That's exactly right.
So really, this is a really fine tool
Yes, it
to
is.
do this.
Right. And we have knowledge about how genes, how we can decipher that code of a
Uh-huh.
gene, and we can understand how that code is used to form a protein. We have we have intimate knowledge of all this, deep understanding, and so our changes that we make are not random, or they're not, crazy. We're not just trial and error. We know exactly by going in and changing these bases or removing these base pairs, we know what we're going to create in the protein that's encoded in that new engineered gene.
Okay, this is fascinating. Okay, Alan. So why would somebody want to do genetic editing? What could be the benefit?
Well, that it's a $64,000 question, right? Because um, we can we can change our world, right? We can change the quality and the quantity of the foods that we need. For example, we can make the foods that we have more nutritious than they are. For example, there's efforts now to in the genetic engineering world to increase the vitamin content of some of the vegetables that we eat. Okay, so not just making um, crops benefits for farmers to make greater crops a yield per acre, but also to improve the quality of the foods that we're trying to generate. And of course, we're increasing the number of people on this earth very rapidly, and it's a serious thing. We all need to be using everything we have now to assure that we have a safe and secure food supply. So that would be one really obvious big reason for doing it.
Uh-huh. What about medicine, health?
Absolutely. They're using, for example, plants to produce some of the medicines that we need. Um, it's cheaper and faster. Faster could be, for example, vaccines. Did you get your flu shot already?
Yes, I
Okay.
did.
Well, you think about it. Every year we have to get a flu shot, and why is that? Because the kinds of flus strains that are prominent each year changes, and so we have to rapidly adapt each year, and we're guessing which vaccine do we need to make. In plants, for example, you can make those vaccines much faster. So there's another example of why you would engineer a plant for human benefit.
What about diseases like AIDS, for instance?
Well, there are many diseases that from our studies we know are, I hate to say the word simple, but have simple solutions. Some are very complicated. That might, for example, might require tens or dozens of genes, but some of them have their origin of disease in a single gene. I can give you, for example, age, as you just mentioned. Cystic fibrosis is another one. We know it's a simple base pair that's altered in people who have cystic fibrosis. And if we can change that base pair, those people would be cured of the disease of cystic fibrosis. Another example of where gene editing would be useful is hemophilia. Again, it's a single base pairing that's different in those people who have hemophilia. If we could go in and edit that gene, we can cure an individual with hemophilia. Back to AIDS, because it's big in the news right now. We just heard 2 weeks ago at the Genome Conference where a scientist in China, Professor He, actually edited the gene of twins, and it was a gene that encodes for a protein. We started out talking about proteins. This protein is on the surface of our cells, and the virus, the AIDS virus, HIV, recognizes that protein at a certain spot, and that enables it to enter the cell. Mm-hmm. What Professor He did was to edit out that little spot on the protein. And by doing so, he didn't change the property of the working property of the protein. He simply changed it so the virus can no longer recognize it, and therefore there are and we know that because there are certain individuals on Earth who are resistant to HIV, and it's because of their missing that particular spot on the protein. And so he replicated that using his genetic tool.
Cool. Well, Alan, this sounds pretty exciting, but I mean, I'm sure one thing on everybody's mind is the possibility for malfeasance. Mm-hmm. This sort of thing can be misused, I'm sure, or it could be intentional or even unintentionally misused. So, yeah. So what are the safeguards here?
Well, let's start with the last, say, 3 decades of this type of precise genetic engineering that we've been doing. It's been moving so quickly that governments and bodies that oversee the work of scientists are trying to keep up, and it varies by country on what's regulated. So in the United States, we have regulatory bodies that try to make sure that what is being engineered is safe for humans or safe to feed to cattle or that sort of thing. But as I said, things are moving so fast that it's hard for these governments to keep up, all right? When it gets to humans, it's a real can of worms, right? We've mentioned some severe diseases, some very severe diseases, hemophilia, cystic fibrosis, for example. AIDS is a disease that comes from our susceptibility to the HIV virus. We might all agree, if we can cure those, then that might be a good thing, right? But what about height, right? And I think we're very, very close to being able to control the size of humans. That's one or few genes which we could edit, you know. And there's no really control, and that's why it's important for all of us to have this conversation right now about what it is that we think is permitted, what's ethical, and what our future is going to be in this area. And we need to be realistic too.
Yeah, I can see how ethics is a can of worms here, but I can imagine even bigger can of worms. And that is, what happens to a population of humans, say, that can affect their evolution? I mean,
yeah,
the population is able to affect the direction of its evolution.
Yeah. Well, we can make predictions because this is an area that's well studied, right? Population genetics is something that we've been studying for a long time, including humans, and we know what happens when certain types of changes occur in the genes of a
Uh-huh.
population. And so we can actually begin to make predictions about what could happen to humans if we're not careful in the way we apply our genetic editing in the future.
Yeah, I guess I may be a little more concerned. If people can, be self-referential, that is, they can affect themselves, uh, then who knows uh, what the survivability of the population is. I guess that's.
Yeah, and what you didn't mention is it's not just at any one point do we want a bigger, smarter, faster human. Everybody wants the best for their children, and imagine now you can actually control that. But on what's going on in the background, of course, we hear a lot about climate change, for
Uh,
example. What is the future? I mean, what kind of humans do we need 100 years from now? What if we don't need to have legs, for example? Right? Maybe
we need fins.
we need fins. Yeah. What if legs actually are bad to have? Right. So as we begin to manipulate human genes, we need to be careful. And I think one big lesson we've learned for sure, and that is diversity. Diversity is really, really important, right?
Uh,
The more diverse, the more diverse set of genes we begin with, the more likely that we're going to have some combination of genes or types of genes that will allow at least a few of us to survive whatever is ahead of us. Okay.
okay, well, thank you, Alan. This has been very enlightening. I certainly didn't know all this before today,
Yeah.
and I appreciate your help on this. And we have a few questions that I think will occupy people for some time to come.
Yeah. You a lot of topics that we've raised today that really need in-depth conversation, not just between you and me, but all of us together.
All of us, that's right.
Yeah.
Policymakers, everybody. All right, well, this is Roger Jones. I'm signing off, still from downtown Pensacola. Until next time, bye bye.
Bye bye.